Okay, so this is the second presentation that is a bit theoretical to give you an overview of what we do in Bgee. And after the break, that will be more practical, where we will show you how to use our tools and retrieve our data. So here I'm going to present how we do the integration of multiple experiments, multiple data sets in multiple species, which are the approach we use to do that. And so I will start from, I will start by using single cell data as an example, because this is the most fashionable data type currently in gene expression. And very briefly, so most of you probably are aware of how single cell data are generated, but just as a reminder, we have beads to which are attached molecules with a PCR primer, a cell barcode, a molecular identifier, and then each cell go through a microfluidic device to be isolated, and then they can be either isolated with a bead, as here, and then there will be a gene expression quantification that can happen for that cell, or sometimes the cell will end up in an empty droplet, so it won't work. But the important point is that here we don't know a priori, we don't know beforehand what are the condition, the cell that we're going to study. So in bulk RNA-Seq data, you sample an organ and you already know precisely what you are sampling. And with single cell data, droplet-based data, it's different in that you don't know beforehand what you are sequencing. So it is a big change regarding curation and data integration. If I give here an example, so you will isolate your cells, you will perform bioinformatics analysis, and you will end up with a clustering of your cells. So each dot is one cell, and it's based on the gene expression quantification. You perform a clustering, and you identify different cell populations. And then you're going to look at the expression of genes in these cell populations, and you're going to try to find genes that are highly specific of one cell population. So, for instance, this gene here, it is expressed in almost all cells, so it has a low specificity. And this gene here is very specific to this cell population, so it's probably a good marker of that cell population to identify what are those cells after the analysis. And at the end, then the author, they will provide annotation of each cell population where they try to identify what those cells were doing. So in all of these steps, it's really an added complexity as compared to bulk RNA-Seq data regarding annotation. So you need to retrieve the raw data with the barcode files, and then many steps are processed, like you filter cells, you filter genes, and you normalize, and you do a dimensional reduction. Then you do a clustering and a differential expression. And finally, at the end, looking at the function of those genes, you are able to provide an information about the cell type for each of your cell population. And so at each of these steps, the parameters are gonna to impact your end result. So this is very much different as compared to bulk RNA-Seq data. So continuing, at the end, what you want to see at the end, you want to see this clustering, and then you can see here on the left, those are the cell type information as provided by the authors. And you can see that those are free text information. So if you want to integrate data between different experiments, you need to standardize those terms so that you know which cell types were the same between different experiments. And here you can see that everything is free text. So you won't be able to integrate between different experiments just retrieving this information. And then you want to display gene expression over this different cell population. And then again, for this, you need to normalize the information between different experiments if you want to provide this information in a very integrated way. So so the challenges we have regarding single cell data, first, you need to get information about the protocol used, because whether it's single nuclei or single cell, you will have unprocessed or mature mRNA, and you won't process the data in the same way. Then you need to get information about the barcode position in the mRNA molecule, and that will change between protocols. So you need to be very careful about what exact version of the protocol was used. Then the annotation is going to be highly dependent on all your processing steps. So for instance, in the fly cell atlas, they provide 2 clusterings as a result. If you go to Fly Cell Atlas and try to download the data, they will provide 2 results. So they provide the result from a stringent clustering and the result from a broad clustering, because it is very difficult actually it requires to manually tailor the result to get the best information. And we found cells that were annotated as ovary cell in the stringent clustering and as neuron in the broad clustering. And those were the experts who generated the data. So you can see the level of complexity that these new types of data bring when you try to integrate everything in a very standardized way. And then again, when we try to retrieve the necessary information, sometimes we don't have access to the barcode file that allow to identify each cells. Then we cannot find the association between barcode and the annotations of the clusters. And even the BAM of FASTQ files, again, I don't want to criticize because it's very complex, but when we try to retrieve the data for the Fly Cell Atlas, the FASTQ files that you find on SRA were corrupted. They were not usable. So we had to contact them so that they could re-upload the data. So you can see that at each of these stage, this is the work we do at Bgee to check everything, make sure everything is available so that at the end, when you go on the Bgee website and you click the download buttons, everything is usable. And now I'm going to show you how we do this integration. So the first level of information we want to integrate in Bgee is where are genes expressed in a very clear way. We want to give a definitive answer, yes, this gene is actively expressed here or not. And in the spirit, it looks very similar to what we can do with in situ hybridization data. So here, this is a picture of a zebrafish embryo, which is transparent. And using in situ hybridization data, you highlight the domain of expression of a specific gene. And this is the kind of very clear answer we want to give in Bgee, but from any data types and in any species, making that comparable. So for doing that, so why would we like to detect signal of active expression? First, like in differential expression analysis, for instance, there is often a first step of filtering the non-expressed genes, the genes that receive 0 count or that are so lowly expressed that you cannot really distinguish their gene expression variability. So it is often a first step in transcriptomics analysis to filter genes that are not actively expressed. Or again, also you might want to compare genes that have an expression that is condition restricted. For instance, sex-biased genes. If you perform a differential expression analysis after removing the genes that are lowly expressed and you want to compare male-female, then you won't be able to compare the genes that are expressed really only in one of the sexes. So it is interesting to have this level of information or of binary information. This gene is expressed in male, it is not expressed in female. And finally, just for answering the question, you are interested in the specific genes, where is this gene expressed? So you will have varying level of expression in hundreds of conditions. Where do you put the threshold to say, this is where this gene is important? And this is the information that Bgee can provide. So how would you do that to determine whether a gene is actively expressed or not? Because you want to know that the gene is expressed over the stochastic noise of the cellular machinery, and you want also to know that your gene is expressed over the technical noise, which can mean that you will have some expression value for genes that are actually not expressed because of, for instance, a technical noise in your experiments. So I will give you an example using RNA-Seq data and starting with bulk RNA-Seq data because it's simpler. So yeah, usually authors in publication to detect genes that are actively expressed, they define an arbitrary cutoff on their data. And that might be maybe 10 counts or 2 TPM or 1 CPM. And what we see in Bgee data is that there is a large variability in the gene expression values between different experiments, and probably it's unlikely that there is one threshold that could match, that could work for all experiments in all different conditions. And again, when you look at publication, there is little consensus on what value you should use for such threshold. So yeah, so in RNA-Seq data, as I said, usually author define an arbitrary threshold at 2 TPM. This is what I see in most of the cases. But here I show you, so we use like a gold standard data set, really telling us in that condition, here are the genes that are really expressed. And we took the gene expression data and checked the true positive and false positive rates depending on different cutoffs. So here you get the true positive rates of how good you were at identifying the genes that are truly expressed, and the false positive rates, so genes that you identified as truly expressed but were not actually. And you can see that, yeah, at varying cutoffs, like 0.5 TPM or 1 TPM, 2 TPM, it's going to be a balance between the true positive and false positive rates, of course. And you can see that at 2 TPM, you achieve in that specific data set only a good balance between the true positive rate here and the false positive rate. So this default threshold that is used in most cases, it's pretty good, but for that one data set. And when we see in Bgee data that it doesn't work for all data sets. Sometimes it has to be 5 TPM, sometimes it has to be 0.5 TPM. So how do we make an adaptive threshold that can adjust to the variability between different experiments? So what we see in bulk RNA-Seq data is that if you do a density plot based on the level of expression, that comes from this quite old now publication from 2011. Here you see a density plot for different type of genomic features. So you get the exonic features here, the intron expression level, and the intergenic expression levels. And what these authors propose here is that it's very typical, this distribution of exonic expression level, where you see here a shoulder on the left of the distribution. This is really very typical. And the authors here propose that it is actually the sum of two distributions, the lowly expressed genes and the highly expressed genes. And they show that this distribution of what they call lowly expressed genes, they show all features of being genes that are not actively transcribed. When you look at the chromatin state, when you look at histone modification markers, you can see that really these genes on the left, they are genes that are not actively expressed, but still they did receive some reads, they did receive some gene expression levels. And what we see is that the distribution of these non-expressed genes, well, they match closely the distribution of intergenic reads. So again, intergenic regions, they are not supposed to be expressed, but because of the stochastic noise of the cellular machinery and because of the experimental noise, these regions, they still receive some reads. So we started from there thinking, okay, reads receive like expression level of intergenic regions, it is a good estimate of this background noise. So that will be our baseline. You answer, a lot of you answer, you need to compare to a baseline. We're going to use the intergenic regions to define this baseline of expression. So basically what we do in Bgee is that we compare the gene expression TPM value to the distribution of the intergenic gene expression level. So when we compare the TPM value to that distribution, we can obtain a p-value. And that's going to be really the added value of Bgee, that for each gene, we can provide you a p-value of whether it is actively expressed or not. So this is the method we are using right now. And in Bgee, we are trying at improving that method. So your baseline is going to be very dependent on how do you define your intergenic regions. So right now, we see that the best result is that if we select intergenic regions that are close to genes, because maybe they are in regions of open chromatin, right? They need to be accessible to the cellular machinery. If you use intergenic regions that are absolutely not accessible, it's not going to give you a good baseline of noise. And then, yeah, you need to know how are you going to represent this distribution of intergenic expression levels? Should you use a normal distribution or a negative bimonial? You know, like, so we benchmark all of those aspects to come up with one statistical method, allowing you for each of your gene or in each of your sample to get a p-value to know whether your gene is actually expressed. So now focusing more on single-cell RNA-seq data. So I showed you, like in bulk RNA-seq, we have this nice distribution of expression levels for genes, for intergenic regions. How do that work in single-cell data? Can we still apply the same method in single-cell data? So here I show you the density plot for a full-length single-cell experiment, so a smart-seq experiment. Which is not like a 3' AN base. You can really sequence the full length of the genes. You can see that the distribution doesn't look as nice, right? So here in red are the protein coding genes, and in blue are the intergenic regions. So even though you don't have this nice shoulder on the left, you can see still that there is a point where these two distribution crosses, and it really looked like, yeah, over the intergenic noise, you can identify the genes that are actively expressed. So it still does work for the smart-seq data because we do have like still a lot of reads, but there is a lot of dropouts in single-cell data. And here I show you like the percentage of protein coding genes that are detected as actively expressed or not in bulk RNA-seq data or in single-cell data. So this is based on our data in human and in mouse. And you can see that from single-cell data, only above like more than 25% of the genes, we are able to detect them as actively expressed, while it should be 80% in human, you know, like. So, and here again, I show you like in two libraries, one that is bulk RNA-seq. So that has been, bulk RNA-seq. You get like a lot of reads, and only 10% of the genes receive 0 reads. While in single-cell data, almost 60% of the genes receive 0 reads. So here we do have a limitation. But again, using this intergenic background expression level, this is very adaptive to your condition, to your experiment, to your data type, so that when we tell you, okay, this gene is expressed, you can be very confident, and we can give you a p-value to integrate in your statistical analysis. So it does still work, even though we have a high dropout. So here,
quick question.
yeah.
Sorry. Yeah. Do you, on the single cell level, do you consider each cell a small bulk experiment, or do you do a pseudo bulk of the whole experiment to determine the p-value and those values?
Yeah, so we do a pseudo bulk. So for smart-seq data, we don't have this problem because one library is one cell. So then really it's the same. But so yeah, I come here to droplet-based data, and so I show here the result for one cell. And for one cell, you cannot distinguish. Basically, everything is stacked on top of each other. So that the current states in Bgee that we cannot do it per cell, but we just very recently tested the method with imputation, where you use method to kind of fill the gaps of the dropout. And actually, when we use imputation, it does work really, really well. So we can use our method to infer the state of expression in each cell when you use imputation. And we compare to a gold standard, and it does work really well. We had a very high true positive rate, a low false positive rate. So it's not yet available in Bgee. Maybe by the next release, we will provide these calls per cell and not only per cell population. So, but yeah, in the current version of Bgee, we hadn't used imputation at that point, and we concluded it cannot be used because all distribution just stack on top of each other. But if we do pseudo bulk, so we aggregate the data for each cell population, then we have more signal. And here again, you have like the intergenic regions. Here you have the genic signal, and you can see that, yeah, you can even see like kind of a shoulder here again on the left of the gene that are not expressed and the gene that are expressed. So again, we compare that to gold standard reference data set to estimate the true positive, false positive rates. And it does work really well, even on droplet-based data, as long as you pseudo-bulk the data per cell population, or if you do imputation to look at each cell individually. But in the current release of Bgee, we didn't use imputation. So that's what we do in the current release of Bgee, that we pseudo-bulk the cell population, we call genes present or absent, and then we go back to the individual cells. It means that if in one cell, if in the cell population we say that gene is expressed, then for all the cells of that population that receive at least one read, we say, okay, that read is for real. It's not spurious data. This gene is really expressed in that cell. Okay, so that's the point, that in Bgee, then we really integrate everything together. So how do we do that? So yeah, first, like for all data types, our aim is to provide a p-value. So for each gene in each condition, the information we can integrate is a p-value. And for each data type, we define methods to do that. So for bulk RNA-Seq, so it's going to be based on the z-score. We look at the distribution of intergenic regions, and then we compute a z-score for your genes, the expression level of a gene compared to that distribution of intergenic expression levels, and we can provide a p-value. For full-length single-cell data, smart-seq data, it is the same, because, yeah, we do have like still millions of reads per cell. And for droplet-based data, such as 10x target-based data, we pseudo-bulk the information per cell population to have enough signal. But we do the same in Bgee. You have also Affymetrix data or in situ hybridization data. So we also provide a p-value. So for Affymetrix data, it's based on the Wilcoxon test of the prop set signal versus the background signal on all the chip. For in situ hybridization data, basically we use information density. So in model organism databases, the more important an expression of a gene is, the more report of that information you have. So basically, it's based on information density. We're going to transform that into a score that we can transform them into a p-value. So yeah, really for all this information, all this data type, at the end, we get a p-value. But then how do we reconcile all this information and integrate based on different level of annotation? Do you look at expression at the brain or at the cerebellum level? How do we reconcile this different level of organization then? So what we do in Bgee is that we use what we call a graph of conditions. So it is a bit complicated at the end of this theoretical presentation, just so that you have an idea of how we do. So Mark mentioned that we use ontology. So we use the Huberon anotypical ontology, where you can have, for instance, the endocrine and exocrine pancreas that are both part of pancreas. So this is a little part of the ontology. But we also have ontology for developmental stage, so that you know, for instance, that an 80-year-old human is part of the age stage when you want to compare aging, for instance. So basically, each sample we annotate to these ontologies. So it means that from experiments for each gene, we're going to be able to say, okay, in that experiment, this gene, we show that it is expressed in that condition. And then we build a graph of conditions. So this is what you see here. We can say, okay, pancreas at sexually mature stage, or exocrine pancreas at fully formed stage. And then we're going to converge. At the end, all those information, they come from pancreas at the fully formed stage. Like it means like after embryo, basically. So you see that we have this varying level of annotation. Here I have like 5. But they can all converge at some point to a same level of information. Okay, pancreas adult, let's say. So we have this p-value of information, and we get basically propagate this information from each experiment. We're gonna propagate those p-values using this graph of conditions, so that at some points, you see, we had information from for different genes A, B, C, that were not comparable. We had information only for some of those genes in these different conditions, but by propagating this information, at some points, we can manage to have information for all these genes in a comparable way. So that's what we're going to do. Propagate individual information in this graph of condition so that at this point, it's going to converge and be comparable. So all these p-values are going to be propagated like that. And then we're going to correct for multiple testing and give you one answer. Okay, for your 3 genes of interest, this is the state in pancreas. Okay, so yeah, so basically we use that very large graph of condition to propagate all the p-values for each gene and each sample. And at the end, we integrate everything by doing an FDR correction. Okay, so it means that we start from hundreds of samples. And at the end, for your gene, we're going to give you one answer in that condition. Yes, gene A is expressed in pancreas with a corrected p-value of 10 minus 3, okay? And that's going to be what we present at the end. And after the break, we're going to show you how we leverage that information presented to you on the website. And then we have various criteria to simplify for you, like so that you can say, okay, please give me only the gene expressed, or please give me only the gene that are not expressed. We summarize this FDR p-value in different category of quality, like present gold, present silver. So this is an information you can find on the website. Considering the time, I will not insist on that. Okay, so here I presented only qualitative information, genes that are expressed or not, but in most studies, you are interested in the quantitative. You want to compare the expression levels. So how do we do that in Bgee to compare the expression levels? So it's based on non-parametric statistics, okay? So we basically use the ranks of genes based on the expression levels, and we normalize these ranks to provide a quantitative information. So yeah, basically for each data type, we rank the genes and we weight these ranks based on the quality of the data, like how good was the data set at identifying gene expression levels. So of course, in situ hybridization data, you don't have information about expression level, as with bulk RNA-Seq data, you do have. So basically, we weight those ranks based on the quality of the information, and then we normalize those ranks between all the data types and all the conditions. And at the end, what you end up with is an expression score going from 0 to 100 maximum. So Mark showed you in the previous presentation insulin in type B pancreatic cell, and that received an expression score of 100. That was the highest expressed genes in that condition. And this you can compare between species and between experiments and between genes based on this non-parametric statistics. So of course, with non-parametric statistics, you do lose some information, of course, but the advantage is that then everything is comparable, everything integrated, again, with that idea that when you go to Bgee, you will get one single answer about the level of expression of your genes of interest. Yeah. So yeah, oh yeah, you're right. So and just to give you an example, so APOC1 is an adipoprotein important in liver. So when you go to the gene page in human for that gene, you see that at the top, we correctly identify liver at the most important condition for that gene with a very high expression. Again, the maximum is 100. So this is very, very high. Then you can see the gene that are the conditions that are less expressed with less expression. But what I want to show you is that how it is comparable between species. If you retrieve the APOC1 gene expression in other species, you can see that for mouse, we also identify liver with a very high expression score. The same in zebrafish, same in those primates, in guinea pig. You can see that consistently the expression score is so high in all the species, and every time we identify at the top condition for that gene liver. And again, like showing even more species, you can see the expression score every time almost 100, and every time we identify liver at the top condition for that gene. So that's how in Bgee we integrate information and then make that comparable between species. And then you can look at the conservation of expression between species. So here I entered the list of APOC1 genes in mammals, I think, And you can see that, again, liver comes up at the most conserved expression, like we have, like in all those species, 30 genes showing consistent expression in liver. So yeah, basically that's what we are a bit late, so I stop here. Yeah. So
yeah. In the chat, there was a question: is the NGS library preparation covering intergenic region?
No, it should not. Usually, you select RNA molecules using poly A tail. You should not have information about intergenic regions, but it is the case that we do. And it's like in RNA velocity analysis, you should not have unprocessed mRNA, for instance. You should not be able to read molecules that are not spliced, but you do. So that's part of the noise of the techniques. I mean. There is also an influence from the protocol of the amount of intergenic expression levels. Yes, it is true that based on the different protocols, it is more noisy. You will find much more reads mapped to intergenic regions, but from what you see, from what we see, it also reflects on the noise of gene expression. I mean, from what we see, the protocols that generate high expression of intergenic regions, it's because they are highly noisy, and it does impact the gene expression as well. Is it not your